Hello guys, welcome to Molecular Biology series. I'm Vineet and today I'll be explaining about the DNA supercoiling. So DNA supercoiling, it actually represents the amount of twists in the DNA. So that means whenever you uh, coil or twist a DNA to further, that develops kind of secondary superstructures which are normally referred to as the supercoils. Okay, suppose consider an example where you have a thread. So where the one end of this thread is fixed to some point and one end is free to rotate. Okay, and at the free end, so whenever you try to pull the threads in opposite directions, okay, one thread in downward direction and one thread in upward direction, okay, whenever you try to pull these two threads in opposite directions, okay, so this kind of pulling actually result in production of some kind of strain on the some kind of strain on the thread. Okay, so that strain is actually relieved by undergoing some kind of extra structures known as the supercoils. Okay, so these supercoils, they try to they are due to this excessive strain on the relaxed thread. Okay, so the same kind of knots or so the same kind of supercoils are also developed in the case of this DNA. Okay, so whenever we introduce some kind of extra twist in the DNA, or whenever we try to increase helical turns in a DNA sequences, okay, so the DNA develops some kind of super secondary structures we often called as a supercoils. Okay, so the supercoiling is the major property for this bacterial chromosomes, because these bacterial chromosomes are in circular forms. Okay, they are the circular chromosomes. Okay, so these circular chromosomes, because they don't have any kind of free ends, okay, as the linear sequences do. So these linear sequences have the free ends, okay, 5 prime and okay, so these are what free ends. Okay, so bacterial chromosomes doesn't have any kind of free ends. Okay, so because of this unavailability of this free ends, they don't have any kind of opportunity to relieve the strain on the DNA molecule. Okay, so because they don't relieve the DNA strain, because they don't relieve the strain on the DNA, they undergo supercoiling. Okay, supercoiling is a essential property for this bacteria. Okay, because the size of bacterial DNA is 1,000 times greater than the size of a bacterial cell. Okay, so if you consider this uh, as a bacterial cell, this circle as a bacterial cell, the size of this bacterial cell is in this range. Okay, so the bacterial DNA will be in this range. Okay, that means approximately 1,000 times greater. Okay, and if you want to fit this bacterial DNA into this simple bacterial cell, for the first you have to compact the DNA by introducing some kind of supercoils. Okay, so these supercoils first compact the DNA into this size, and then by attaching some kind of scaffold proteins or the nucleotide associated proteins, which we call them as the naps. Okay, so these naps, they further condense the DNA and make the DNA into a compact size molecule, and now it could be able to fit into this bacterial cell. Okay, so this is what the size of the bacteria, and this is what the size of the actual DNA molecule when it is present in the relaxed state. Okay, so this is what the supercoiling plays a major role in compacting the DNA for the bacterial cell. Okay, it not only plays a major role in the bacteria but also in the eukaryotic cells, where the transcription and the replication process going on. Okay, so whenever this transcription or the replication process happens, the helicase molecule, which is involved in the separating of this DNA strands. Okay, so these helical molecules, when it runs in 5' to 3' directions, okay, in the forward directions, okay, it introduces some kind of strain on the DNA molecule in forwarding chain. So this kind of strain actually makes the DNA to overwound itself, making the DNA to have a positively supercoiled DNA. Okay, so this is what the positively supercoiled DNA is. Okay, so this kind of introducing some kind of extra helical turns, okay, or some kind of introducing some kind of extra twists on the DNA molecule causes the overwinding of the DNA, which we often refer to as the positive supercoiling. Okay, so this positive supercoiling is mainly due to the strain on the DNA molecule that is produced by the helicase enzyme, which runs from 5' to 3' direction. Okay, and in the back of this replication fork or the transcription fork. Okay, so this is what called as the transcription fork, which runs in forward direction. Okay, so back of this transcription fork, the whatever the DNA sequences are there, they are subjected to underwinding. Okay, so this kind of underwinding creates the negative supercoiling. Okay, so this kind of underwinding create the negative supercoiling in the DNA, and we often refer this DNA as the underwound DNA. Okay, so this is what happens whenever the replication fork or the transcription fork runs in 5' to 3' direction or the forward directions. Okay, but in the case of this replication we won't be seeing any kind of negative supercoiling, but we will be seeing only formation of the precatenates. Okay, precatenates are nothing but some kind of extra knots on the DNA molecule. Okay, whenever there is a coiling of two DNA daughter molecules occurs. Okay, so we will be seeing this kind of precatenates. Okay, and in the replications, okay, in the forward direction where the helicase moves in 5' to 3' directions, okay, we can only see this positive supercoilings happening. Okay, so these are what the positive supercoilings are, and these are mainly due to the overwinding of the DNA. Okay, and in the transcription we can see this negative supercoiling as well as the positive supercoils. Okay, at the back of this transcription fork, this is what the transcription fork is. Okay, so when this transcription fork moves in forward direction, positive supercoils are introduced on the DNA, and at back of this transcription fork the negative supercoils are introduced on the DNA, so that makes the underwinding of the DNA. Okay, so these supercoils are due to underwinding. Okay, and these supercoils are due to what? Overwounding. Okay, okay, so what we have seen in initial experiment where you fixed a simple thread into fix a simple thread to one point, and the other point is which is free to rotate, and you whenever you try to pull the threads in opposite directions, that gives some kind of extra stress on the molecule to develop some kind of super secondary structures. Okay, the same thing happens over here. Okay, so whenever you are moving your helicase in forward directions, okay, it actually uh, separates the DNA strands in opposite directions. Okay, so this kind of pulling, okay, actually develops a lot of strain on the DNA molecules. Okay, so to compensate this kind of strain on DNA molecule, the DNA overwinds itself, or further coils itself to make the DNA to look like as a supercoiled DNA. Okay, so this is what about the supercoils. Okay, and these precatenates in the replication forks, they are mainly due to the formation of supercoil or coiling of two separate daughter DNAs, one upon each other. Okay, so this is one daughter DNA and this is one daughter DNA, which are the products of the actual replication. Okay, so when these two daughter DNA molecules, when they coil over each other, they produce some kind of secondary structure, super secondary structures called as the precatenates. Okay, we often refer to them as a quarter DNA duplexes. Okay, so that means we have four DNA strands. Okay, so these four DNA strands are what referred to as the quarter DNA, and since they are forming a simple duplex, okay, we can also call them as a quarter DNA duplex. Okay, so this statements actually describes about this processes or the supercoiling things happening in the replication and the transcription. You can even see this during the DNA replication. Precatenin is found behind the replication fork. Okay, so precatenin is mainly due to newly synthesized duplexes wrapping around one and other. Okay, and in the case of this transcription process, we can see this positive supercoiling happening in the forward direction of the helicase or the advancing protein machinery. Okay, so in front of this advancing protein machinery, we will be seeing this positive supercoils, and in the back of this advancing protein machinery, we will be seeing this negative supercoils. Okay, and the negative supercoilings are mainly due to the underwinding. Okay, so this is whatever the basics of this supercoiling, and as I've said you, the positive supercoiling is actually due to underwinding. Okay, so overwinding. So whenever you overwound the DNA, okay, so that DNA undergoes positive supercoiling. Okay, suppose consider you have a DNA molecule over here. Okay, so whenever you introduce some kind of extra helical turns, or whenever you try to twist this DNA, this is actually a relaxed DNA or the BDNA. Okay, normal BDNA. Okay, whenever you try to twist this BDNA or introduce some kind of extra helical turns, or overwind this DNA, it undergoes positive supercoiling. Okay, like this. Okay, so here the helical turns are what, 1 2 3 helical turns. Suppose we have 3 helical turns in BDNA. Here we have 1 2 3 4 and 5. So extra, we have 2 extra helical turns. Okay, so this kind of increasing the helical turns on a normal DNA actually, makes the DNA to undergo positive supercoiling, because this is the positively supercoiled DNA, which is mainly due to overwinding. Okay, and whenever you try to further relax this BDNA, already relaxed DNA, so whenever you try to further relax, or whenever you try to do underwounding thing, okay, that results in the negative supercoiling, okay, or producing a negatively supercoiled DNA. Okay, so and this DNA, which is formed due to underwinding, okay, so now contains less number of helical turns. Okay, that means, so this is what, the actual 3 helical turns, now got reduced to 2. Okay, so this kind of decreasing the helical turns, or doing underwinding to the already relaxed DNA, makes the DNA to undergo negative supercoiling. Okay, so this is what called as a negatively supercoiled DNA, this one. Okay, so this negatively supercoiled DNA actually resembles the structure of ZDNA. Okay, so this ZDNA is actually a slim and elongated DNA, or elongated form of the BDNA. Okay, so this one, and this overwounded DNA, or the positively supercoiled DNA, actually resemble the structure of ADNA. Okay, so these are what, different forms of the DNA that are formed due to supercoilings. Okay, so this is what supercoiling creates. Okay, so this supercoiling mainly creates a tension on the DNA. Okay, and whenever the DNA contains free ends, it can relieve the strain, or it can relieve the tension that is produced on the DNA. If the DNA does not contain any kind of free ends, then it is subjected to supercoiling. Okay, so that is what I gave in this point. Okay, so this point actually tells you supercoiling creates a tension in the DNA, and thus can only occur in the DNA that has no free ends. Okay, that means, it should be there in the circular form. Okay, otherwise free ends can rotate to relieve the tension. Okay, so the linear DNA sequences like this. Okay, so these linear DNA sequences are the free ends. Okay, and they have the opportunity to relieve the tension. Or in linear DNA, if it is anchored to a protein scaffold. Okay, that means, if you look at the eukaryotic genome, so these eukaryotic genomes, they contains long threads of the DNA molecule, which are attached with some kind of histone protein, some kind of non-histone protein. So they are called as the condensin and the cohesin proteins. Okay, so these condensin and cohesin proteins, they attach to the DNA molecules. Okay, at some specific points. Okay, and they act as barriers for this DNA sequences. Okay, they act as ends of the DNA sequences. Okay, so, or fixed ends of the DNA sequences. Okay, so since they are acting as a fixed ends for the DNA sequences, they can allow the DNA molecule to undergo supercoiling. Okay, so in the eukaryotic chromosomes, where there is a huge amount of the genome, okay, so we can observe this supercoiling. Okay, but in the case of short DNA sequences, like, around 1000 base pairs or the 2000 base pairs, which this DNA sequence having free ends, we cannot observe this supercoiling. Okay, because they have the free ends to relieve the tension on the DNA molecule. Okay, so this is what about the positive and the negative supercoiling. And if we talk about the circular molecule, because it doesn't have any kind of free ends, so it cannot reduce the strain, so it is subjected to supercoiling. Okay, and underwinding a linear DNA presents no problem, because the ends are free to spin and can able to relieve the strain on the DNA molecule. Okay, even if you introduce some kind of twisting, it reduce the strain on the DNA sequences. Okay, in a typical chromosome, as I've said you, in case of eukaryotic genomes, okay, so they contain a lot of condensin proteins or the coexin proteins, which are fixed to the DNA sequences, which make the DNA to get compacted. Okay, so these proteins actually act as barriers or the ends of the DNA sequences, so they might introduce the supercoilings on the DNAs. Okay, so underwinding of a right-handed helix, so just remember this one, underwinding of a right-handed DNA helix also facilitate the formation of short stretches of left-handed ZDNA. Okay, where the base sequence is consistent with the Z form. Okay, so that means, whenever you underwound this BDNA, so that BDNA forms the ZDNA. Okay, so this ZDNA is actually a slimmer and elongated version of the BDNA. Okay, so this, if you want to convert this BDNA, just you have to do the underwinding thing. Okay, so supercoiling also changes the overall topology. That means, overall deformity of this BDNA. Okay, suppose consider you have a linear DNA sequence of 20 turns. Okay, and when you try to link the ends of the linear DNA with other ends, okay, like this, and try to circularize the linear DNA into circular DNA. Okay, so now this circular DNA also contains the 20 turns. Okay, so you are just circularizing the linear DNA of 20 turns, okay, to make it into a circular form. Okay, so this circular form now also contains the 20 turns. Okay, so the same terms which are present in the linear DNA. Okay, and if you introduce underbonding, or if you remove two helical turns from this linear DNA, okay, so it can compensate the strain on the DNA molecule. That means the linear DNA, since it has the free ends, okay, it can compensate the underbonding or the supercoiling thing. Okay, but in the case of the circular DNA, uh, the compensation thing will not be happening, because it doesn't contain any kind of free ends. Okay, because of this, it will now only has 18 turns. Okay, and this is what the tensioned state. Okay, so now the circular DNA is in the tensioned state. Okay, so in order to get into a stabilized state, okay, so this will undergo supercoiling, okay, in this form, which actually contains two super helical turns. Okay, these are what called as the super helical turns. Okay, and 20 helical turns. Okay, the helical turns are again stabilized. Okay, so this is what the unstable structure of the DNA whenever you try to introduce underbonding or the overbonding. So this is actually underbonding happening on the DNA molecule. Okay, so whenever the underbonding happens, this molecule tries to get stabilized. Okay, so this stabilization forms the supercoiled structures with two super helical turns over here and two helical turns on the DNA molecule. Okay, so this kind of supercoiled DNA is actually present in the case of this bacterial cells. Okay, so this is what, and this supercoiled DNA is actually energetically favored form of the DNA, where this one, or the newly introduced newly underbonded DNA, is unstable form, or energetically less favored form. Okay, and in the biology, energetically favored form or favored form molecules only exist, not the energetically unfavored molecules. Okay, so if we talk about the conformations, okay, so the adaptations of this supercoiled DNA is different for different different types of DNAs. Okay, so DNA also present in uh, DNA also has different handedness. Okay, that means the DNA can be in the left-handed form or in the right-handed form. Okay, so this kind of handedness makes the DNA to undergo different types of supercoilings. Okay, to adapt different type of structures in when it is subject to supercoiling. Okay, so there are two major possibilities for this DNA to adapt when it undergoes the supercoiling. Okay, so one is the plectronomic supercoiling, or the interbonding supercoiled DNA, interbond supercoiled DNA, or the toroidal supercoiling, or the solenoidal model of the DNA. Okay, so in the plectronomic supercoiling, whenever you try to introduce the supercoiling in middle of the DNA, that means whenever you try to interwound the DNA in middle, suppose this is the DNA sequence which you have, and whenever you try to induce some kind of extra helical turns in middle of the DNA in this part. Okay, so this causes the interbonding, and this makes the DNA to develop the plectronomic supercoiling. Okay, so this kind of plectronomic supercoiling is actually seen in the case of twisted ropes. Okay, so we have seen the first example during the start of the lecture. So that kind of supercoiling is actually called as the twisted rope. Okay, and plectronomic supercoiling is mainly seen in the bacterial genomes. Okay, where they have circular DNAs. Okay, or the circular chromosomes. Okay, so we can see this plectronomic supercoiling. Okay, and interwinding mainly happens in the case of this circular chromosomes only, and not in the case of this linear chromosomes. Okay, plectronomic supercoiling, it helps in compacting the DNA, making it more manageable within the cell. Okay, plectronomic supercoiling also crucial for the process, also crucial during the process like DNA replication and transcription, where it helps unwinding of the DNA helix. Okay, so in the case of this bacterial replication, and also in the for the transcription process. Okay, this plectronomic supercoiling helps the DNA to undergo itself, and making it readily available for the lot of replication machineries or the transcription machineries. Okay, So that is what plectronomic supercoiling plays. How does this plectronomic supercoiling maintain the structure and topology of the DNA inside the bacterial cell? Okay, and whenever you look this plectronomic supercoiling or the plectronomically supercoiled DNA under the microscope, okay, so it appears as a series of intertwined loops or branches. Okay, that means so in this form. Okay, so the whenever you look at this plectronomic supercoiled DNA, it appears in this form, or the series of branches or loops, okay, intertwined loops. Okay, and if you talk about this toroidal supercoiling, which is mainly seen in the case of this eukaryotic genomes, okay, where the solenoidal mode of the chromatin material is due to this kind of toroidal supercoiling. Okay, so in case of this eukaryotic genomes, the long DNA gets wrapped over the histone core proteins. Okay, so the nucleus to form the nucleosome structure. Okay, so this nucleosome structure actually represents the toroidal supercoiling. Okay, DNA wraps around a protein core, forming a spiral toroidal shape. Okay, spiral or toroidal shapes, which we also call as solenoidal model. Okay, and toroidal supercoiling is essential for the higher order packing of DNA in eukaryotic cell. Okay, it allows efficient organization of the large amounts of the DNA within confined spaces of the nucleus. Okay, and if we observe this toroidal supercoiling under the microscope, okay, it actually appears as series of loops or coils. Okay, so this plectronomic actually appears as intertwined loops or branches, but this toroidal supercoiling appears as series of loops or coils, okay, around the central axis. Okay, that means there is some kind of a central axis over here. Okay, and on this central axis you will be seeing DNA getting wrapped over this central axis like this. Okay, so this kind of supercoiling is also called as a toroidal supercoiling, and this kind of supercoiling is also called as a plectronomic supercoiling. Okay, this is what called plectronomic, and this is what called as a toroidal. Okay, so this is the major difference between the toroidal and the supercoiling. Okay, and plectronomic you will only be seeing in these bacterial chromosomes where the DNA sequences are in the form of circular molecules. Okay, and the toroidal supercoiling will be seen in the case of eukaryotic genome where this genome is in a linear form. Okay, and has to get undergo high order packaging in the eukaryotic nucleus. Okay, so this is what the diagrams of this plectronomic and the toroidal supercoilings. Okay, so this is what a simple nucleosome structure is. Okay, so in the central axis you will be having a lot of histone proteins. Okay, so these histones are like basically basic amino acids. Okay, contains the basic amino acids in them, or the positively charged amino acids. Okay, so these positively charged amino acids wrap the DNA, the negatively charged DNA over themselves. Okay, so that forms a toroidal coiling, or a kind of solenoid model. Okay, and the same DNA, so whenever you try to introduce twist in mid of this DNA, in mid section of the DNA, in this part. Okay, so this circular DNA, okay, so due to this interwindings undergoes plectronomic supercoiling. Okay, so this is in the case of bacterial plasmids. Okay, so that means the helical turn or the pitch angle is quite reduced. Okay, and the bend angles will be very high in the case of these bacterial chromosomes. Okay, so that means uh, you are further compacting the DNA to make this DNA into a broader form of the normal version. Okay, so this is actually this actually resembles the A form DNA. Okay, if you are aware of this A form, B form, and Z form of the DNA, okay, you will be knowing it easily. Okay, so this is actually represent the A form DNA. Okay, so this is what the normal DNA, relaxed DNA. Whenever you try to interwound the DNA, it undergoes plectronomic supercoiling. Okay, and that is mainly seen in the case of bacterial plasmids. Okay, and if you look at the properties of this toroidal supercoiling and the bacterial plectronomic supercoiling or the bacterial supercoiling, this toroidal supercoiling actually has high bend angles. Okay, bend angles due to presence of this the bend angles due to wrapping of DNA sequences over the histone core protein or in the nucleosomes. Okay, these are actually high angle bends. Okay, and then we have the negative rate and high negative rate and high height twist. Okay, which I'll be discussing in this lecture only. Okay, in front, and if we look at this plectronomic supercoiling, okay, it actually has low pitch angle whenever you if because this is because you are compressing the DNA molecules, okay, from either sides, that causes the low pitch angle, and also we have high bend angles on the bacterial chromosomes, okay. So this high bend angles mainly seen when when different type of scaffold proteins attached to the DNA molecules, okay. These are what the some kind of extra proteins which binds to the supercoiled DNA, and this kind of binding actually makes the bend angles to be high, okay. And in the case of these eukaryotes, these are the solenoidal models. These are actually a simple nucleosomes, okay. These are nucleosomes, okay. So when these nucleosomes they try to form a higher order packing, okay, so we can observe this toroidal supercoils, okay. Toroidal supercoils, mainly due to presence of a central axis, okay. So you will be seeing a central axis in the case of this toroidal, but not in the case of this bacterial supercoils, or plectronomic supercoils, okay. And then the handedness of the supercoils, okay. I've already discussed about the handedness of the supercoils, okay, based on the handedness property, or based on the type of DNA, whether it is a left-handed DNA or the right-handed DNA, the property also changes, okay. The adaptations also changes, okay, whether it would be a toroidal form or the plectronime form, okay. Suppose if you have a left-handed DNA, left-handed helix, okay, DNA in the left-handed form, okay, and if you introduce the negative supercoilings to that DNA, okay, so this left-handed DNA will be in the toroid form, okay. So that means in the solenoid, what we have seen in the case of the solenoid model, the round form, okay. So this left-handed DNA, whenever it undergoes negative supercoiling, will form the toroidal supercoiled DNA, okay. And the same DNA, the left-handed DNA, okay, if you introduce positive supercoiling, okay, so that undergoes to form a plectroneme supercoiled DNA, okay. So the plectroneme structures for the left-handed DNA helix are mainly due to the positive supercoiling, okay, and the toroidal structures for the left-handed DNA helix are mainly due to the introduction of negative supercoiling, okay. And the same goes for this positive right-handed helix also, but in the opposite way, okay. For the right-handed DNA helix, okay, whenever you try to introduce negative supercoiling, okay, it develops the plectronemes, and the same right-handed DNA helix, when you try to introduce the positive supercoiling, it develops the toroids, okay. The same thing happens, okay, the toroids and the plectronemes, okay, both DNAs, both right-handed and the left-handed DNAs adapt the toroid forms and the plectroneme forms, but the supercoiling is different in the case of this toroid and the plectroneme forms of the right-handed and left-handed DNA, okay. So if it is negatively supercoiling, or in the case of negatively supercoiled DNA, left-handed DNA has two toroid forms, and right-handed DNA will be having the plectronemes, okay. Similarly, the positively supercoiling, in the case of positively supercoiled DNA, the right-handed DNA helix will adopt the toroid forms, and left-handed DNA helix will adopt the plectroneme structures, okay. So this is what the handedness of the supercoil differs in different supercoiling mechanisms, okay. And in the case of humans, we have condensing proteins, okay, a large protein complex, okay. So this plays central role in mitotic chromosome assembly, where the mitotic two daughter chromosomes, after synthesizing the DNA in S phase, okay, whenever they enter into the mitosis, okay, mitosis phase, okay. So this formed two daughter chromosomes, they get attached, okay, and with the help of this condensing proteins, okay. So these condensing proteins, in the daughter chromosomes, okay, in the mitotic chromosomes, they induce the supercoils, okay, by using some kind of ATP molecules as energy batteries, okay. And in the case of this bacterial species, we have lot of similar kind of proteins or scaffold proteins, which we often call as a nucleoid associated proteins, or NAPs, okay. I've already said you about the NAPs, okay. So in these bacterial cells, okay, so there are certain type of barriers, okay. So the barriers to supercoiled diffusion divide the genome into series of topologically isolated supercoiled domains, okay. So the long DNA is actually divided into specific domains, okay, which we often call as a supercoiled domains, okay. And these supercoiled domains, okay, so majorly involved in the organizing the nucleode, okay, entire genome of the bacterial species, okay. And these supercoiled domains, they are negatively supercoiled, okay, and they also they are associated with the nucleoid associated proteins, okay, or NAPs, okay, so which further organizes the bacterial genome into a compact sized molecule, okay. And if you look at this nucleotide associated proteins in the maps. Okay, so they are called as the DPs. Okay, DPs are nothing but the DNA binding proteins from starved cells. Okay, so these are actually observed in the E. coli cells. Okay, which has been shown to bind supercoiled DNA much more rapidly than torsionally reacted DNA. Okay, so they bind to the supercoiled DNA and they readily compact the DNA, to make the DNA to be fit in the bacterial cells. Okay, so this is what about the handedness of the supercoiled DNA. Okay, I've said you right, the toroidal form may also attain the prectonomic form, and the prectonomic form of DNA may also attain the toroidal form of the DNA. Okay, so if you look at this nucleosomal structure. Okay, so this nucleosomes, they form the solenoid model when they try to attain the next level packing in the organism of the chromosomes. Okay, whenever you try to remove this nucleosome structure, okay, simple nucleosome core particle, okay, from the DNA, where the DNA is wrapped around this nucleosome core. Okay, so whenever you remove this nucleosome core, this free DNA, where the left DNA between the two nucleosome cores. Okay, so this now adopts the plectonym structure, okay, or plectonomic structure. Okay, so this kind of nucleosomal removal or the histone core removal, okay, convert the solenoidal form of DNA into plectonomic structure. Okay, so we can also make the solenoidal DNA to get converted into plectonomic structure. Okay, and the same thing happens in the case of this what the bacterial DNA, but this is a very rare case. Okay, the NAPs or nucleotide associated proteins, majorly binds to the plectonyms or the supercoiled DNAs rapidly, and this rapid binding causes them to compact more uh, to get fit into these bacterial cells. Okay, and they also allow the lot of different transcription machineries and the replication machineries to perform their actions on the DNA molecules. Okay, so this enhancing ability for replication process or the transcription process is maintained by the nuclear associated proteins. Okay, so this is what about the supercoiling, the different types of supercoiling, so the adaptations of the supercoiled DNA. Okay, so then we have the linking number. Okay, so this linking number is actually defined as the number of times a second strand intersect the surface. Okay, that means consider you have one strand DNA over here. Okay, so DNA strand over here. Okay, so this is first DNA strand, and the second DNA strand. Okay, so whenever this both DNA strands they try to link. Okay, they develops certain type of a topological bond. Okay, so which is entirely different from this base stacking interactions and the hydrogen bondings. Okay, so this topological bond, okay, so the newly developed bond which is mainly due to the interaction of these two separate DNA strands. Okay, so this kind of bond actually enhances the stability of the DNA. Okay, so enhances the physical nature of the DNA. Okay, to be more stabilized form. Okay, whenever you try to remove these hydrogen bonds and as well as the base stacking interactions, okay, this topological bonds still remains intact. Okay, so that is what I've said you in this second point. Okay, if two strands are linked as shown in figure, they are effectively joined by what we described as a topological bond. Okay, so this hypothetical bond or an imaginable bond we also called as a topological bond. Okay, if we even if even all hydrogen bonds and base stacking interactions were abolished, okay, that means if we remove all the hydrogen bonds and the base stacking interactions, okay, this such that the strands were not in physical contrast, okay, this topological bond would still link the two strands. Okay, that means this bond actually enhances the stability. Okay, and if we even remove all the necessary interactions for the DNA strands, okay, this topological bond even plays a major role to maintain the stabilized structure of this DNA molecule, or to maintain the links of this DNA molecule to be in the helical form. Okay, so this is what the linking number is. The linking number is which is a topological property. Okay, it only changes when you cut the DNA sequences and unwind the DNA sequences. Okay, but it does not changes whenever you bend the DNA sequence, or whenever you deform the DNA sequences. Okay, or the linked DNA sequences. Okay, so this is what the linking number is. Okay, and linking number is defined with the symbol LK. Okay, so and this linking number is majorly for the circular DNAs only, closure circular DNAs. Okay, which we often call as a covalently closure circular DNAs. Okay, cccDNA. Okay, so this covalently closed circular DNA can only have a linking number but not the linear DNAs. Okay, and linking number could be positive or negative. Okay, and positive sign is given for the interwinding in a right-handed DNA helix. Okay, and negative sign is given for the interwinding for a left-handed helix DNA. Okay, so for the right-handed DNA helix, if we perform the interwindings, okay, so we would often refer them, we would often sign them as a positive. Okay, define them as a positive. And if we interwound the left-handed DNA helix, okay, we often define them as a negative. Okay, so this is about the linking number. Okay, linking number could be negative or positive. Okay, if we remove the helical turns, okay, so that causes the linking number to be in the negative sign. Okay, if we introduce some kind of extra helical turns, that makes the linking number to positive. Okay, the same thing happens over here. Suppose you have a linking number 200. Okay, so 200 links are there in this normal DNA. Okay, so this is the consider this is the bDNA only, like normal DNA. Okay, so this initial bDNA has the initial linking number of 200. Okay, so that means LK naught will be the 200. Okay, if you break the strand, okay, so if you create nick over here, okay, that means you are actually unaffecting the linking number. Okay, so if you are not changing the linking number, okay, that means you are actually relieving the strain on the DNA molecule or the supercoiling molecule. You don't have you're not uh, changing the, you are not actually changing the linking number, but actually changing the topology of the DNA. Okay, so when you change the topology of the DNA, the linking number does not apply to that DNA molecule. Okay, so DNA linking number is actually a topological property of the DNA. Okay, which only depends on the the nicking properties. Okay, like whenever you try to cut open the DNA sequences, and whenever you try to change the supercoiling things, okay, so that causes the linking number to be uh, diminished. Okay, but the linking number does not changes if you introduce some kind of extra turns, or if you bend the DNAs, or yeah, linking number changes whenever you try to increase the helical turns. Okay, whenever you open the helical turns, or whenever you introduce the strain on the DNA molecule. Okay, but it does not changes whenever you bend the DNAs, or whenever you introduce some kind of deformities. Okay, so whenever you try to remove the helical turns from the DNA, okay, the linking number changes. Okay, suppose if you are removing two helical turns from the normal relaxed DNA, so the linking number changes to what? 198. Okay, so the difference between initial linking number and the final linking number will be -2. Okay, so this negative sign indicates the DNA is in the left-handed helix form, left-handed form, or negatively supercoiled DNA. Okay, so that is what. Okay, so if the linking number is in the minus or negative sign, that gives the negative supercoils. Okay, that means removal of two helical turns. Okay, if the linking number is +2 or in the positive sign, okay, that is called as a positive supercoils, or if we add +2 supercoils, okay, +2 helical turns, that will be the linking number will be 2, not 2. Okay, it gets increased. Okay, the positive term indicates the positive supercoils. Okay, and the negative term indicates the negative supercoils. Okay, this is very simple. Okay, and if you break the strand, linking number will be diminished, and there will be no such property defined the linking number. Okay, because you are removing the because you are cutting the DNA sequences. Okay, there is another term called as a superhelical density, which actually relies on this linking number. Okay, so this superhelical density is a measure of degree of supercoiling a DNA molecule, or it can be a measure of number of turns removed relative to the number of number present in the relaxed DNA. Okay, so how many turns are removed on the normal DNA molecules. Okay, from the actual turns which are present in the relaxed DNA. So that measures the superhelical density. Okay, so how to calculate the superhelical density, which is mainly read on the linking number. Okay, so first, if you want to calculate the linking number, the superhelical density, first you have to know about the initial linking number. Okay, the formula for the initial linking number is what? LK naught. Okay, so this LK naught is actually given by the base pairs in the DNA sequences, the entire base pairs in the base pairs in the DNA sequences, divided by 10.4. Okay, so this 10.4 is taken as normal. So this 10.4 is actually for the bDNA, or the relaxed DNA. Okay, since all the DNAs will be in the, since majority of the DNAs will be in B form only, or the relaxed form. Okay, so we actually consider the normal denominator as 10.4 only. Okay, so this actually represents the BDN. Okay, so this base pairs will be the total number of base pairs that are present in the circular chromosomes. Okay, so this actually gives the linking number, initial linking number. So then you can find out the superhelical density. Okay, suppose we have a problem over here. Okay, so what is the superhelical density of a closed circular DNA with length of 4200 base pairs? Okay, so this DNA has 4200 base pairs and it is in the closed circular form. Okay, and a linking number of 374. Okay, so oh yeah, its linking number, initial linking number is what? 374. Okay, what is the superhelical density of same DNA when linking number is 412? Okay, so whenever you increase the linking number to 412, so what will be the superhelical density of this DNA? So that means you are either removing or either introducing some kind of extra turns. Okay, so which also changes superhelical density. Okay, and are these molecules negatively supercoiled or positive? You have to tell the adaptation of this DNA molecule. Okay, whether it is the negative or positively supercoiled. Okay, first we have to do this. First we have to calculate the linking number, initial linking number. So that is I have already given the base pairs upon 10 power 4. Okay, so that base pairs were what? 4200. The value of the total length of the DNA, closed circular DNA, is 4200 base pairs. Okay, if we divide this, if I divide the length of DNA by 10.4 10.4 base pairs per turn. Okay, so we actually get the value around 400. Okay, and by using the changing linking number, that is the what we got is the, so initial linking number will be 400. Okay, so the actual linking number is what? We have 374 over here. Okay, so the change in linking number will be minus 26. Okay, this minus sign already indicates that is in the negatively supercoiled form. Okay, or the left-handed DNA, and substituting the values, the delta LK and LK naught, into the formula, superhelical density formula, we actually get this value. Okay, minus 0.065. This minus sign actually indicates the DNA is negatively supercoiled form. Okay, and suppose if we increase the linking number to 4112, as I said in the question. Okay, so when the same DNA has a linking number of 412, the change in linking number will be 412 minus 400. That actually that actually gives the plus 12. Okay, so this plus sign, plus 12 or normal 12, okay, it actually indicates the positively supercoiled DNA. Okay, so from the superhelical density formula, okay, or the signs which indicate the linking number or superhelical density, you can actually determine how the DNA is supercoiled. Okay, whether it is a negative supercoiled, how the DNA is okay, whether it is a negatively supercoiled DNA or the positively supercoiled DNA. Okay, and you can also determine the superhelical density. Okay, then we have different properties for this linking number. Okay, so linking number actually contains two structural components, twist and the wedge. Okay, twist is what? The simple thing, the normal definition for the twist is the number of helical turns in one strand. Okay, so how, if you twist the DNA, that increases the twisting number. If you remove the twist, or if you remove the helical turns, that decreases the twisting number. Okay, that is very simple. Okay, and wraith is what type of a confusing property of this linking number. Okay, it actually refers to the coiling of DNA helix itself. Okay, that means we have seen the superhelical coils in the normal DNA. Whenever the double stranded DNA undergoes superhelical turns, okay, like this. Okay, so these are called as the superhelical coils. Okay, or superhelical linking numbers. Okay, superhelical links. Okay, so these actually represent the wraith. Okay, so wraith refers to the coiling of DNA helix itself, such as supercoiling. Okay, and the linking number formula will be, if we talk about the twist and wraith, okay, if you link the twist and wraith with the linking number, then it will be the formula will be twist plus wraith. Okay, so this is actually a confusing topic. Okay, I might not focusing much more into this. Okay, then we have the topoisomerases, which catalyzes the changes in the linking number. Okay, so they actually cut the DNA sequences and unwind the DNA sequences, or relieve the strain on the DNA sequences. Okay, So they do this unwinding or relieving the strain on the DNA molecule by making some kind of transient covalent bondings. Okay, so topoisomerase 1 and topoisomerase 2. Okay, type 2 and type 1 topoisomerase and type 2 topoisomerase. They form a transient covalent bond with the DNA sequences, and they cut the DNA sequences, and they relieve the strain on the DNA molecule. Okay, by using a strand passing mechanisms, or by actually rejoining the broken ends, by passing one more strand from the actual ends. Okay, so this is what they do, and they eventually change the linking numbers by increments of 1 or 2. Okay, if the topoisomerase 1 if they act on the DNA sequences, they change the linking number by 1. Okay, if the type 2 acts on the DNA molecules, and if they break the DNA molecules, and they change the linking number by 2. Okay, so this is what about type 1 and topoisomerase. They are further classified into different categories. Okay, in type 1 topoisomerase, we have three types. Okay, subdivided into 1A, 1B, and 1C. 1A, it forms a transient covalent bond with the 5' DNA phosphate, while 1B and 1C, they form the transient covalent bond with the 3' DNA phosphate. Okay, and in the case of this type 2 topoisomerase, they also form the transient covalent bond with the 5' DNA phosphate. Okay, both of the subtypes. Okay, like type 2A and type 2B, they form the transient covalent bond with the 5' substance only. But the main difference is what, in the subunits of the protein molecules. Okay, type 2 topoisomerase, type 2A topoisomerase actually contains uh, two monomers. Okay, two different monomers. Okay, while type 2B contains only one monomer, which will be involving in entire mechanism process. Okay, so this is what about the classification is, and I've also given a lot of mechanisms of actions of this topoisomerase. Okay, and the species which contains this kind of topoisomerase. Okay, so if you look at this type 1 topoisomerase, it is actually subdivided into type 1A and type 1B, type 1C. Okay, the main difference between this type 1A and type 1B is the formation of transient covalent bond. Okay, so type 1A forms a transient covalent bond with the 5' phosphate, while type 1B and type 1C forms the transient covalent bond with the 3' phosphate. Okay, and the type 2B, this contains two monomers, one monomer and another monomer. Okay, heterodimer. Okay, it forms the type 2A forms the heterodimer. Okay, but binds with the 5' phosphate only as a transient covalent bond. Okay, type 2B contains a simple monomer. Okay, which is involving in the entire mechanisms. Okay, and these are the actual roles for this topoisomerase. Okay, so major one is the bacterial DNA topoisomerase 1, bacterial DNA topoisomerase 3, and we have a DNA reverse garase over here. So these DNA garase, okay, it actually introduces the single stranded break. Okay, and mainly present in the sulfolobus acidocaldaracea. Okay, so this is what the bacterial DNA garases do. Okay, they belong to the type 1A subfamily, type 1A topoisomerase, and induce single stranded breaks in the DNA molecule to relieve the strain on the DNA molecules. Okay, and we have a lot of mammalian topoisomerase type 2 alpha, type 2 beta, which introduce the double stranded breaks and pass the DNA strand from the another strand. Okay, to relieve the strain on the DNA molecule. Okay, so this is what about the different type of topoisomerase, and if you look at the function, uh, the actual mechanisms. Okay, so this is what they do. The supercoiled DNA. Okay, so they type 1 introduces one strand break, one strand cut, or one strand break to relieve the strain, or to change the linking number by 1. Okay, so that means if the linking number is 3, the initial linking number is 3 for this supercoiled DNA, it will be changed to linking number, it will be changed to 2. Okay, LK2. Okay, and if the type 2 topoisomerase, if they act on the supercoiled DNA, the linking number is changed by increments of 2. That means initially is change in linking number will be 3 minus 2, so the change in the LK will be what, 1. Okay, so the circular chromosome will now not have any kind of super helical turns. Okay, and if you look at the mechanism, so this type 1 topoisomerase, which forms a transient covalent bond with the 5' phosphate. Okay, so we have a 5 carbon over here. Okay, of the ribose sugar. Okay, this 5' carbon, or the fifth carbon atom of this ribose sugar contain the phosphate molecule. So this phosphate molecule, forms the bond with this tyrosine molecules. Okay, whatever the lone pairs are present, whatever the lone pair present on the oxygen molecule, first forms a imaginary bond with the phosphate molecules, and then form the actual bond. Okay, so this bond is actually called the transient covalent bond because it further breaks into uh, two parts. Okay, so this kind of bond breaking, this kind of bond formation actually breaks the bond between the above base and the below base. Okay, this one, and this forms a single stranded cut on the DNA sequences. Okay, and after passing the after passing the other strand from this break like this. Okay, so it actually relieve the strain on the DNA molecule. Okay, by passing the unbroken strand through this broken strand. Okay, and after doing all this thing, the lone pairs which are present on this oxygen or atom, the above base, okay, they again form a bond with this phosphate molecule, imaginary bond, and then form the complete structure. Okay, like this phosphodiester linkage. Okay, so this kind of a median interaction. Okay, median interaction of this topoisomerase 1s with the topoisomerase 1 enzymes with the DNA sequences, and forming a transient covalent bond. Okay, actually reduces the strain, or the linking number of the DNA helix. Okay, the supercoiling thing of a DNA helix. Okay, so remember that this type 1 topoisomerase, they contain the tyrosine in their active site, and this tyrosine atom contains hydroxyl group in the R chain. So this hydroxyl group can only form the covalent bonding with this phosphate molecule, 5' phosphate molecules of this DNA sequences. Okay, and then only it can release the linking number. And if you look at the type 2 topoisomerases, the type 2 topoisomerase alpha, which is a heterodimer, okay, which also contains the N gate and the C gates. Okay, so they first interact with the DNA sequences, which has to get double stranded cuts. Okay, then the N gate also interacts with the normal DNA, the second DNA sequences, second DNA strand. Okay, and after inducing a cut by using ATP molecules. Okay, it uses two ATP molecules to one ATP for cutting the DNA strand. Okay, introducing the double stranded breaks. Okay, and one ATP for passing the DNA strands into the unbroken DNA strand through the broken DNA strands. Okay, so this passing actually changes the linking number on the DNA, and the two DNA molecule will now be left by this topoisomerase 2 alphas. Okay, so this is what the major mechanism in the case of this